I'm Abigail Calder from the Research and Knowledge Exchange Department here at the Mind Foundation. And I'm very happy to introduce to you today Ula Kozlowska. She's a postdoc at the Institute of Bioorganic Chemistry at the Polish Academy of Sciences. And her work investigates psychedelic tryptamines as therapeutic candidates for helping with inflammation and boosting regeneration in damaged brain tissue. I'm very excited for this. Please give her a warm welcome.
Check, check. Can you hear me, right? Okay. So my background is in the research on the neurodegeneration and transplant immunology, and I found that the psychedelics have some features that may be interesting to treat the neurodegenerative disorders. So this is like my first try to research that. So here, from psychiatry to the neurology, psychedelics as candidates to provide brain protection from the neuroinflammation. So this is the plan of the presentation. There will be a comprehensive introduction on the brain pathology, the biology of microglia, because I was making the research on microglia cells, and the psychedelics as immune factors, and the materials and methods and the results and the conclusion. Please tell me, hands up, how many of you have the background with the immunology, like on the academic level? Cool, this is so good. That's a relief. Okay, but there also are people who are not familiar with it, so I will have to try to handle it okay. So, in the aging society, we have some struggles to treat the brain diseases because the brain is a very fragile and inaccessible organ. Each year, we have a problem that the stroke takes 15 million of people each year worldwide. 5 million of those are fatal cases. Another 5 million suffer severe disability. So this is the brain damage. And with the neurodegenerative disorders, the dementia disorders, 10 million diagnosed annually. 50 to 70% are the Alzheimer disease. And the number is going to be doubled by 2040. Another example, motor neuron disease. I have the background from this disease from my past projects. So this is like the rare disease, but also the number of the patients suffering this disorder gonna grow with the age, with the time. So there is no cure currently available for the brain diseases, but as the psychedelics made some promises in the psychiatry, maybe, just maybe, they can be the next candidates for therapeutic molecules in the neurodegenerative disorders. So this is the model, this is the explanation of the neuropathology in the neurodegenerative disorders. So we have some gene mutation, which then creates, those genes creates the misfolded protein, which are abnormal and dysfunctional, and they get aggregated in the tissue. So here are seeing the ataxin inclusions from my laboratory, but because this is the spinal cerebellar ataxia type 3. But with the like Alzheimer disease, there will be like amyloid beta plaques, or in the motor neuron disease, it would be like SOD protein aggregates, and this will lead to the mitochondrial dysfunction, the oxidative stress, neuroinflammation, neurophagy, blood and brain barrier breakdown, neural cell loss, and the disease symptoms. So, of course, the psychedelics won't help here, because if we have the gene mutation, it can be caused from the familiar factors, but it also can be the environmental factors, or the stress or the pollutions. We can't heal the gene mutation, but the psychedelics may be helping to modify the disease on those levels. So what does the neural tissue look like? So we have the neurons, networks, and we also have the helper cells, like the oligodendrocytes or the astrocytes that help to make the hospitable environment for the neuron to work. But we also have the microglia cells, and you perhaps heard that the microglia are the immune cells in the brain. So microglia can, depending on the signal, can be pro-inflammatory or anti-inflammatory, and make some different functions, but both of those are important. The pro-inflammatory will help to fight the infection, for instance, or to clear those toxic protein aggregates, and the anti-inflammatory will help to restore the tissue, to regenerate the tissue. And it's fine, they need to stay and be regulated with each other. But also the microglia has some different functions. So those related to immunology, like phagocytosis, or the antigen presentation will help to provide the protection from infectious agents or those toxic protein aggregates. But they also take part in the myelinogenesis. This is like making this shelf on the neural cells. They will help in the astrocyte activation and growth. They will help in the neural stem cells differentiation. So from the very beginning, the microglia cells helps or assist the neural stem cell to differentiate. So they are not just the destroyer cells, they are also the nursing cells, the helper cells. And when this neuron gets mature, it will help in the synaptogenesis and synaptic pruning. And also the modulation of the blood and brain barrier, so the microglia may send signal to open the blood and brain barrier, which is normal during the homeostasis. But the problem begins when we have the disease and there is a disbalance, so the microglia gets overactivated, hyperactivated. It will affect all of those functions, but it may lead to the abnormal phagocytosis, and this can kill the neuron. So even the microglia can phagocyte the violet neuron. So the microglia can be cause of the problem, and it happens in the neurodegenerative disorders and the brain damage. It can also activate the astrocytes to open the blood and brain barrier so we can have the leakage, which can be dangerous. And of course, those unhospitable environment, pro-inflammatory environment will affect the neural stem cells and the neurogenesis. Fortunately, psychedelics are anti-inflammatory agents, and we have proofs for that. So this is the excellent study from the Charles Nichols laboratory, when they showed that the serotonin receptor can be the agonist, the deoxymethamphetamine, the agonist of this receptor in vivo will downregulate dose-dependently the pro-inflammatory tissues, pro-inflammatory cytokines in the tissues, like the small intestine or the aortic arch. So this is happening via serotonin receptor, but also with the sigma-1 receptor, the DMT or the 5-MeOxy-DMT in vitro in the monocyte derived dendritic cells, which are kind of similar to the microglia cells. This downregulated the pro-inflammatory cytokines in vitro via sigma-1 receptor. But what about the microglia cells? So the aim of the study was the investigation on how the DMT and the psilocin will influence the microglia biology in vitro. So I performed two experiments. One was the phenotype evaluation after the activation of the microglia cells, and the second was the in vitro co-cultures with the neural cells. So I made this neural monolayer and that put the activated or not activated microglia cells with or without the psychedelics and see what happened. So this is the plan of this study. So I isolated the cells from the young mice. Then after I got the microglia culture, I added the vehicle media, which does nothing, But the LPS, I will explain later, will activate the microglia cells. And after 12 hours of incubation, I added this DMT or the psilocin and performed the test. The immunofluorescence cytometry or those co-cultures experiment. So this is the model. So I really want you to understand the model and why I studied those markers, like the TLR4 or NK-kappa beta. So the toll-like receptor is like a sensor on the microglia cell that will sense the bacteria, but more particularly the bacterial LPS, which is the lipopolysaccharide. So if the toll-like receptor reacts to the LPS, it will start this very complicated cascade that will lead to the activation of another factor I was researching, the NF-kappa beta, that's induced the pro-inflammatory response. So, for instance, the secretion of the pro-inflammatory cytokines. And then it will, this reaction with the LPS will upregulate the CD80, which is like it will provide the co-stimulatory signal, so it's crucial to higher the adaptive immune response to the place, like the T cells. And those are the pro-inflammatory markers. And this one is like anti-inflammatory marker, the TREM2, which is called triggering receptors expressed on myeloid cells too. So this is it. It's theoretically, this is like being researched recently, this is like a hot topic right now. It will provide the neuroprotection and anti-inflammation. So is it clear so far? Of course it's not. So I created like the different slides for people that are not familiar with the biology, the cellular biology or the immunology. So let's start from the very beginning. We have the toll-like receptor, which is the sensor of the bacteria LPS, which is the fragment of the bacteria called lipopolysaccharide, the LPS. And this reaction will make the microglia very angry. So it gets pro-inflammatory, right? And when it gets angry, it grabs the weapons. So, and will call other cells. So the NF-kappa beta that will lead to the secretion of the pro-inflammatory cytokines. This is like those weapons and those tools to inform the other cells that we have the intruder. And then the CD80. This is the co-stimulatory signal. So this is like hiring the professional killer here. So this is the T cell, and this is the professional killer. So seek and destroy the intruder. Yes, okay, I will do it. So this is also the inform others. And the anti-inflammatory, so this is the pro-inflammatory signal, of course. And the anti-inflammatory signal, this is like, protect the neuron. So via TREM2, which reacts to the, also the LPS or the amyloid beta, it will help to protect the neuron. So for instance, the abnormalities in this expression is related to the Alzheimer disease. So now we can get to the results. So at the beginning, I saw that when I treat the microglia cells with the the DMT or the psilocin, when they are activated, see, here are unactivated cells, so they're like that. But if they get angry, they get spiky. They look like they are angry, right? So if we add the DMT or the psilocin, it changed this morphology to more amoeboid, which means that they are activated, but not necessarily in the pro-inflammatory way. So we could measure that with the span ratio. The lowering of the span ratio, the circularity of microglia gets more amoeboid, so it raises up, and the fractal dimension goes down with the DMT. So the TLR, you remember the TLR, this is the sensor of the LPS. It makes the microglia cell get angry, and it gets upregulated. This expression gets higher when there is the LPS. So here there are cells TLR. This is stained in the red, and this is vehicle. So this is the control, and here you can see there is some expression, like higher expression of the TLR4, and you can see also it is on the graph. But if we add the DMT of the psilocin, this level is down. So it's like the control level of the expression of this marker. Now, NF-kappa beta, also the pro-inflammatory signal. It leads to the secretion of the pro-inflammatory cytokines. Here, the control, there is some expression, but not high. And with the LPS, it grows up. You can see it here. But if we use the DMT or the psilocin, it goes down even in the vehicle. So it's like make it down in both activated and not activated microglia. So the CD80, which is for me like especially interesting. Also have a background in the transplant immunology. So this is the flow cytometry result. So this is the histogram when it moves to the right means that the signal is stronger. So here the control, we have some expression, this is normal on those cells. But if we add the LPS, the expression gets stronger. So this histogram moves to the right. And we can see it here. But if we add the DMT of the psilocin in the activated cells, it goes down. And even with the control of the vehicle, the psilocin make it significantly down, and the CD80 down. on the microglia cells. And the TREM2 protective receptor expression, those are a bit problematic results because with the vehicle media, no stimulation, adding the psilocin make it to grow up, the expression of those anti-inflammatory receptor. But in the LPS, somehow DMT prevented it from growing up. So no neuroprotection in this way, like lower, when it comes to the DMT. And this is the second experiment in the co-culture. So remember, I made this monolayer of the cells. Then I added the microglia. It could be not activated or activated. Here's no microglia, so this is like 100% percent of the monolayer. In the control, no killing of the neural cells. So microglia somehow just lived with the neural cells doing nothing. But if this model of hyperactivity of the microglia cells with the LPS made it eat, or I don't know if it was phagocytosis, sorry, I'm not sure it was phagocytosis, but it would make it kill, make it dying of the neurons by 50%. But we could prevent it adding the psilocin, but not the DMT. So we have the two conclusions here. The first one is that the DMT and the psilocin potentially influence the microglia to acquire the pro-regulative and anti-inflammatory phenotype. And the second is, the results suggest potential to use the psychedelics for the treatment of neurodegenerative disorders and brain damage where the microglia and inflammation significantly contribute to the pathology. But it was like very small funding and the small experiments. I didn't answer through which receptor it happens. I don't know what will be happening in the in vivo model because this is just the in vitro model and not in vivo model of the neurodegenerative disease. But fortunately, I got funding. So the public money and Cincinnati grant for young researchers, 1 million zloty, and this is like 200,000 euro, to study the application of psychedelics in the neurodegenerative disorders. This is the spinal cerebellar ataxia type 3, our unique model, mouse model, in the preclinical setting. So I hope maybe in 2 years I'll be able to stand here again and present the exciting results. So I hope right now you love the immunology. And thank you for your attention.
Thank you very much. That was definitely one of the most entertaining microbiology presentations I've ever seen. We do have plenty of time for questions, maybe even a little bit of discussion. So if someone wants to start it off, you will get a microphone. I see you back there.
What's the concentration of the compounds that you used? Maybe I missed it.
Yes, it was. I didn't speak. It was on the slide. I didn't speak. The concentration of the compounds was 100 micromole.
100 micromole.
Yes. It was hard. It was established before by Attila Szabo as effective.
More questions? Yes, right here.
Yeah, basically, I wanted to ask the same question. Would you consider 100 micromolar as a physiological dose?
Yes, this research goes through the peer review, and it was like the same suggestion from the reviewer that it would be better to maybe try more physiological concentrations. And I think I would really like to do it because it is very crucial, I think.
Because as far as I remember, what did you use? 10, yeah.
10 micromolar for the
Or psilocin.
For psilocin, yeah. Right.
Okay, I see another one here in the green shirt. Eric will bring you a microphone.
I remember you showed some results of the small intestine. I was wondering if those cells were actually neurons or other cells.
From the small intestine, it was in the tissue, so it was like in vivo.
Okay, so what I'm wondering here is, can we observe the anti-inflammatory effects of psychedelics only in neurons or also in other tissue?
It was established before, I don't understand.
No, I'm wondering if this is the effect on neurons or if
Here?
this is other cells. Yeah.
Here, no, this is from the tissue. So this is the small intestine here and the aortic arch. So this is like in a heart.
So different tissues. Then.
Yes.
Yeah.
You can check these studies from the church because they have more and they are like really amazing. So.
Okay, I see one more here in the striped shirt. I saw you first.
First of all, thanks for the nice pictures. Even I could understand. I have a question on the duration. Was it 12 hours at 100 micromolar, or was it once this 100 micromolar concentration dosing and then 12 hours later the test?
So here, at the beginning, I added, let's start from the LPS, 150 nanograms per milliliter of the LPS at 24 hours. So it's like 12 hours. It was like 24, like in summary, but after 12 hours, I added the DMT and the psilocin for another 12 hours.
Thank you very much.
Sure.
Okay, here in the back.
Hey, thanks very much for the talk. My question's a bit more broad, and it's about sort of research into epilepsy and psychedelics, and whether you have any thoughts on that or any mechanisms.
Epilepsy?
Yeah.
You know, I think that in epilepsy there is involved the abnormalities in the TREM2 expression, right? So, it's hard, you know, to foresight. It's so far away. But I think that I would like to perform more experiments on the TREM2 because I think that this is like the good direction. And maybe if it really like changes those expressions, like making it up like the psilocin, I think it may be theoretically helpful, right?
Thank you.
I'd like to give some space to our online audience now. Do you want to check if they have any questions in the Q&A section? And while he's checking, I want to tell everybody that our speaker has a speaker's corner right after this. Also for our online audience, it's an event that you can join. So if you have more questions that we don't end up having time for, you can follow her to the speaker's corner and ask them there.
Sure.
What do we got, Eric?
So we have a question from Ulf Bremburg, CEO of Osman Labs. And his question is, your results indicate the immune response to provocation, such as LPS, is reduced. Now, do you see a risk for increased sensitivity to infections in this case?
Yeah, I was thinking about it, and I think it can be a problem. But it's hard to say that we are sure that it will happen like that. But I think that the experiment with the bacteria, for instance, would be very, very important here. It's a very good question.
Thank you.
Is that all we got online?
That's all we've got so far.
All right, so how about right here?
Hi, thanks for the great work and presentation. How does this, it's a very naive question, but how does this compare to other known anti-inflammatories? Like with a lot of drugs that decrease inflammation, why are psychedelics in any way special?
In this situation, this is like, they are not only anti-inflammatory, but they also support the neurogenesis, right? So this is like the two good things that we can give to the patients. The best it would be if it has no hallucinogenic effect, right? In this case of the neurodegenerative disorders, because the people just want to live normal lives, right?
And just a small follow-up, has anything else ever been shown to reduce these sort of inflammatory factors in a similar way?
Could you repeat the question?
Have there ever been other molecules that have gone through the development pipeline that have also decreased these inflammatory markers in a similar way?
Yeah, I think that the maybe average anti-inflammatory factors like the non-steroid anti-inflammatory drugs. Yeah, sorry, I'm already tired.
Thanks very much.
Okay, we've got time for one or two more. Yeah, right here.
Thank you for the presentation. My question is about the outlook of your...
Sorry, sorry, like the cyclosporine and stuff like that. It also
Okay, my question is about the outlook of your future study on the animal model. So, which disease do you plan to model, and how is that going to look like? Are you going to look longitudinally, or just like one dose and then do the test? So, just a little bit more about the animal model that you plan to investigate.
We will use the spinocerebellar taxotype model, and we have like very unique, like only we have this model in our laboratory, and it's not going to be a single dose. It's going to be administered for 6 months weekly. So a lot.
That's very chronic use.
Yes, because this is like this anti-inflammatory response needs to be chronic here. Like to see in this model, which is a very fast model of the neurodegeneration, to see comparing to the control if this is like improvement or not.
Thank you. And I don't know, do you have any comments or knowledge about, for example, effects of psychedelics on, for example, multiple sclerosis or?
Not.
Okay, thank you.
Okay, let's take one last question. Yeah, right there.
Yeah, you talked about neurodegenerative disorders in general. I was specifically interested in rheumatoid arthritis. Do you know anything, maybe either if you're going to look at it or other literature specifically on that?
Yes, so you are speaking about the autoimmunological diseases.
Yeah.
There is one research from the Charles laboratory. He is about the asthma, but there's also, I think this is the review paper by Attila Szabo, and he has about the like prospect applications of psychedelics in the autoimmune diseases. So maybe
yeah,
there.
Can you say the name again?
Attila Szabo.
Okay, thank you.
He's like a very good researcher.
Okay, thank you again, Dr. Kozlowska.